Hello, everyone. My name is Bangju, a research fellow from University of Surrey, UK, and thanks for this great opportunity to share my research on IWA Water Congress. My topic is about deciphering anaerobic ethanol metabolic pathway shaped by operational mode. First, we know that anaerobic digestion, or AD for short, is a sustainable technology in converting organic matters in wastewater into biogas for energy recovery. In AD system, ethanol serve as an important intermediate products. For example, it can participate in three types of methanogenesis. It can improve system stability and enrich geobacter. Basically, there are several metabolic pathways involved in ethanol metabolism. First, ethanol can be converted into acetate and hydrogen via a conventional pathway. And second is it can be converted into acetate and electrons through DIT pathway. And the third pathway is propionate producing pathway with carbon dioxide feeding. So we can raise these two scientific questions. First is how to regulate this pathway, and second is how to improve the reaction rate. We can see that the relevant metabolic pathway occurred in specific functional bacteria. First, geobacter can degrade ethanol into acetate, electron, and protons. Decephalobacter and Pelobacter was the main role in propionate production. Therefore, regulating their enrichment, we can select suitable operation conditions for enriching based on their kinetic parameters. Here is a list of kinetic parameters of EOB. However, there is a lack of data on geobacter in using ethanol. But etc., they found that geobacter also has a low growth rate in ethanol feed system, which makes it as a case strategist. Therefore, we can use RK selection theory to enrich functional bacteria or EOB in different metabolic pathways. Basically, organisms could be separated into R strategists and case strategists. R strategists refer to a non-crowded environment with high growth rate and a reaction rate. And case strategists, they prefer a resource-limited condition with a low growth rate and low reaction rate. So the key factor in regulating RK strategists is food to microorganisms ratio, or FM ratio. In practical application, we could regulate operational modes, SRT, HRT, to modulate the FM ratio in the system. For example, in sequencing batch reactor, or SBR, there is a nutrient gradient. Substrate concentration will decrease with time, and R and K strategies that could be enriched. And the substrate concentration in continuous flow reactor, CFR, is continuously low, which is beneficial to K strategists. Thus, we can regulate operational parameters to enrich specific organisms for the regulate metabolic pathway. The ethanol oxidation process not only requires adjustment to metabolic pathway, but also needs to consider metabolic rates. In addition to changing the operational parameters in the system, the addition of conducting material is also an important way. Well, point-activated carbon has been studied in enhancing the degradation rate of other substrate in AD, and there is a limited research on its use with ethanol. Therefore, we select PAC as conductive material in our research, and we try to solve these three problems. First, we would like to investigate the effect of different operational modes and the addition of PAC on microbial activity and microbial enrichment. And second, we would like to unravel the anaerobic ethanol metabolic pathway and understand the role of carbon dioxide in ethanol metabolism. And third, we would like to examine the contribution of long-term and short-term PAC dosages for enhancing microbial activity. Therefore, 4 reactors were operated in the chapter with 2 operational modes, SBR and CFR, and 2 with PAC addition and 2 or not. And after 6-day operation, 4 reactors were in a steady state. In the long-term result, we found propionate was accumulated in SBR before 34 days, and then totally degraded after 34 days. And I just found the metabolic pathways from KGG database. And we found, okay, there are two possible pathways to generate propionate from ethanol. One is MMC pathway or succinate pathway. Another one is acrylate pathway. And MMC pathway has, carbon dioxide participation. So to confirm which pathway was happening in the system, we just monitored the VFA and methane dynamics in a typical reaction cycle. First, we found that ethanol to propionate pathway remained active in SBRs after 34 days. And second, we found the addition of PAC could enhance the ethanol degradation and reduce the methanogenic lag phase. And third, we found both PAC addition and SBR could promote propionate production and their degradation. Moreover, to evaluate the role of carbon dioxide in the system, we remove carbon dioxide and bicarbonate in the cycle experiment to see how carbon dioxide affects the propionate production. And first, we found that within the initial 2 hours, the degradation of ethanol and the production of propionate and acetate were strongly inhibited without carbon dioxide and bicarbonate. And second, we found that the existence of carbon dioxide could enable the production of propionate in SBRs. And third, we also do a thermodynamic calculation and found that in theory, we need to maintain the carbon dioxide partial pressure over 7.39 atm to make the conversion of ethanol to propionate favorable. Besides, we also evaluate the PAC addition strategy, long-term or short-term, and is that immediately happened or require some time for accumulation? Here are some findings. First, we found that short-term PAC addition could also lead to enhanced propionate degradation and production in SBR reactor without PAC addition. And second, we found that the all ethanol degradation, acetate production, degradation, and all biological reaction rates were increased in CFR with short-term PAC addition. And third, we found PAC addition could not only enhance the conductivity of sludge, but also facilitate electron transfer among various types of species, no matter long-term or short-term. Regarding the microcommunity, we found that CFR and extended SRT could highly enrich G-bacter, which is a electron-active bacteria. And second, the presence of Deciphering could be the main reason for propionate production in SBRs. And we also found that in the ethanol metabolic pathway, the functional genes, we've noticed that acetyl-CoA is a turning point, and carbon dioxide also participates in many reactions in propionate production. And there's only MMC pathway were detected, no acrylate pathway. And regarding the functional genes and their relative abundance under different conditions, we found that the main enzymes were alcohol dehydrogenase and acetaldehyde dehydrogenase. They are the main enzymes in degrading ethanol, and their relative abundance in CFRs were higher. And also we found some enzymes were highly expressed in encoding the conversion from acetyl-CoA to pyruvate and then to propionate, and their relative abundance in SBRs are higher. Moreover, the metagenomic data also shows that the 1.2.3.1 butyric synthesis and all ethanol to propionate related genes in Deciphering in SBRs were higher than that in CFRs. And G-bacter and the related genes in ethanol to acetate conversion were highly enriched. And this figure also shows the roadmap of how to regulate ethanol metabolic pathway with operational mode. Okay, here are some take-home messages. First, the operational mode of SBR, coupled with the existence of carbon dioxide, could facilitate the shift of ethanol metabolism towards propionate production. And second, SBR possesses advantage over CFRs in terms of ethanol degradation and methanogenesis. And third, we found CFR with extended SRT could achieve a remarkable relative abundance to over 70% for G-bacter. And the last is the PSC addition strategy. We found no matter long-term or short-term, PSC addition could both enhance the conductivity and reduce the lag phase in methane production. And also, moreover, the long-term PSC addition could notably improve the rate of ethanol degradation and propionate production and degradation. Well, in the end, I would like to say thank you to the sponsor EPN8 and the CI Founding, and also the University of Surrey and the University of Galway. And if you have interest in my research, feel free to scan my code and keep in touch. Thank you for your attention.